So first, a little background about myself. I come from Mexico. Maybe you can hear it in my accent because people say that to me. So I did a bachelor's degree in marine biology at La Paz, Mexico, and then there I started working with the sharks and rays to study the genetic variability of the samples we found in the fish markets to see if it's really the fish they are selling to us. Then I moved to France to do my master's in biological oceanography and marine ecology, and I focused in the modeling courses. And there I did it's where I started with DEB to study larval mortality. And then I started my PhD. My first year I did it in Brest to work with L'Orpecrie to start working with the DEB model for Sargassum. And then I moved last year here to SET to do my second year and my third year. So what's Sargassum? Holopelagic Sargassum, sorry, it's a brown macroalgae that's invading the tropical Atlantic. Before it was only described in the Sargasso Sea, here more in the north part of the Atlantic Sea. And since 2011, we have found like a big quantity of sargassum that it's now in the tropical Atlantic and the Caribbean Sea. So as you can see here, since 2011, every year we have this event that it's coming into the Atlantic Sea. And for example, this year, sargassum was a record in the Mexican Caribbean. As you have many people that are doing this. And the problem is that it is pelagic, so all his life is in the pelagic way state. But then it strands in the coast of Mexico, of the Caribbean, of Brazil, of Africa, and it causes ecological, economical, and societal problems. To this, we add that we have three different morphotypes that have different forms, that it's Sargassum fleetans, natans, and natans vinge, and then they show spatio-temporal variability. So here I show you a graph. For example, this is also for the Mexican Caribbean, and in 2018, it was Sargassum fleetans that was the most abundant. But in 2020, it was Sargassum natans vinge that was the most abundant. So for now, people that are doing experiments with Sargassum have shown that maybe they have different physiologies, and that, for example, Sargassum fleetans is more resistant to high temperatures, that Sargassum natans has higher photosynthetic capacity, but we have still questions about this. The modeling efforts from now to understand what happened in 2011, to understand the seasonal and internal variability, and also once we understand everything we can predict, have mainly focused on the physical factors. Here is a paper that focused on wind, and in each time point, they use a different wind factor to try to see which one was the best or the better one to reproduce the sargassum trajectory. In black is the trajectory of one sargassum. And there's few studies that have included the biological factor. There's this study of Joanno 2021 that used temperature thresholds and nutrient quotas, and they successfully predicted the seasonal variation of sargassum. However, there's only one paper that has used different parameters for the morphotypes, and they didn't manage to estimate the observed biomass. They estimated a higher biomass than the one we observed. There's not understanding of the interannual variability. So each year we can see that, for example, in summer, this is the period where we have more sargassum, but then to pass to the other year, to the next year, it's we have not been able to study this. And also one experimental study showed that all the studies have focused on only growth rate of sargassum. There's not all the other processes inside of sargassum that has been studied, and then that we need this kind of studies with growth rate, with photosynthesis, to be able to predict sargassum's growth. So this is why my PhD project is biomass. It has a synergetic approach between modeling and experimentation. So my PhD is the work package one. I don't know where is the thing, but my work package is in my PhD, sorry, is the work package 1 and work package 4. And I work in I work with people in Mexico that are doing the lab experiments. And so the objective of my PhD, sorry, is to have like this parallel between lab experiments and numerical experiment. So I do the simulations, and they will do they are doing the lab experiments, and the objective is to compare these two things. So the two objectives of my PhD is to model the individual response of the three morphotypes using a DEB model, and then to integrate it in a dispersion model as a ichthyop to study the sargassum dynamics. So I have told you what is that I'm working with DEB, but maybe you have heard dynamic energy budget, but you don't know maybe or you haven't heard what it is. And it's a mechanistic model to understand the interaction between the environment and the physiology of an organism. So here I show you a fish, for example, and what the model is that it integrates temperature or other bioenergetic factors, food that it's entered the organism. Then it how the organism assimilates it, stocks it in a reserve, how then it is allocated to pay for somatic maintenance, that it's to have the organism living, to create structure that it's growing, and also to allocate it to maturation, or to reproduction. And the importance of this model is that it's based on the energy concepts and the conservation of energy. So it's a model that it can be applicable to many organisms, for example, fish and even sargassum. And in the case of fish or bivalves, for example, we can follow all the life cycle of the organism. But in the case of algae, we do not have only one kind of food that it's entering, but we have different assimilation pathways. This is why we need a multi-research depth model in which we take into account temperature, but also light, for example, and nutrients. And this is the model I'm working on. So we have two reserves, that it's carbon and nitrogen, but the substrates that are entering are carbon, light, nitrogen, and then we follow how the flux are in the organism and how they're allocated to different processes as somatic maintenance and structure. So the first thing I did is to study the different model structures. There's not many papers in algae, but there's 3 papers that have studied the 2 research model for microalgae and 2 for macroalgae. And this is the scheme. And what is easy for this algae is that it doesn't reproduce, so it's only by fragmentation, so we don't have to put the compartment for reproduction, but just to allocation to structure. Then I passed to a 3 research model that they study cyanobacteria, and I tried to do the simulation and try to understand what was happening if I could explain sargassum with a 3 research model. However, as I told you, algae models are really scarce. For example, for the DEB library, there's only 0.3% of papers that talk about algae. And also for the knowledge of sargassum, there's no knowledge about the carbon, nitrogen, phosphorus ratio. So we don't have data to feed our model and tell us something about that. That is why I selected only the 2 research model, and I did the simulations, but we are not using the 3 research model. The next step was to search for the available data to calibrate my parameters. So this is one parallel thing we do. So we have what we call the real world, that is the data on the experimental studies, but I have to translate this into the abstract world, what I can use in my model. So I did the compilation of papers taking into account the initial conditions, what we call observables, and the forcing conditions, and I have 10 available papers. So this is the kind of analysis I made. So I have in the right the lab experiments where I do an analysis of the conditions they use, and then I translate it into what I can actually use in my model. This is the result. So I have the environmental variables or culture conditions, and then I go to the abstract world, what we call forcing variables, and we can see that I have here a steady environmental condition, steady temperature. The only thing that varies is light here. That is because of the photo period. Then I use my depth model with the core parameters, and I do what the experimental people call the model magic. And I have now the simulations. With the auxiliary parameters, I can translate it into observables, which now I can compare with the response variables that are the actual results of the paper. So this is the result after estimation, but it's just an example to show you how it works. So then I asked the question of how many exploitable data I actually have. So the only 3 data sets that I have, it's 2 data sets about the evolution of wet weights according to different temperatures, and 1 curve of photosynthesis data. But I have still missing data about many processes that we know algae do, like nutrient uptake, what happens when we have starvation conditions. We don't have a wide range of temperatures, which is also important to calibrate of our parameters. And for any of this study, we don't have the initial condition of the algae. So we don't know the state of the algae at the beginning of the experience, and we don't know if the results are because of the environmental conditions or because maybe they didn't have enough nutrients in their tissues. So this is like if you don't have enough data, okay, we cannot do anything. So when we don't have enough data with enough environmental conditions, what happens if we do a calibration is the parameters will not have a biological significance. And then when we will try to do predictions, they will not be certain. We cannot do real predictions. So I asked myself, can I estimate informative parameters with the available data? So to this, I create a protocol to test this, in which I create pseudo observations with the available data. So I do simulations, and I estimate my parameters. Then I measure if I can retrieve my initial parameters. And if the answer is no, I cannot retrieve these parameters, then I have to ask myself, okay, what kind of experiment, what kind of data do I need to be able to retrieve my parameters? So that's what I did. I add new experiments that are created by the model. And I recommend, I estimate my parameters, I ask the question. If yes, that means that I can inform now the people, for example, in Mexico that are doing the experiments to say, we need this kind of experiments to understand sargassum physiology. So this is the kind of result we have. As I told you, there's no data on the nitrogen uptake. So when we have no data, this is the error of estimation. We see that the error is bigger than if I have this data. So it's really simple. And with this, I have like a quantitative way of telling people, okay, we need this, please do this experiment. Then I told you about the morphotypes, and this is, so I studied also for the experimental data, what is the response they published about the different morphotypes. And the conclusion is that each study has different conclusion, and it's not easy to actually measure this. For example, here for Sargassum fritans, it's 28 the optimum temperature, that here in this other paper, it's lower temperatures that are the optimal ones. For Sargassum natans vincae, the same. Here it's 22, and here the optimal is 29. So with the data I have, I cannot have information about this. The same goes for the growth rate at different nutrient treatments. So this is between different morphotypes, but when we see the data of Sargassum fritans, they did with different nitrate and nitrogen phosphate and only phosphate, and they also didn't find difference between treatments. And in the case of the photosynthetic capacity, the two data sets that the new papers that we have, they don't have the same conclusion. Here for Sargassum fritans, they say, and natans, they say that there's a difference, but in the below, they said that there's no difference. And the only thing they both of them said is that Sargassum natans, here if you see at 35 degrees, photosynthesis is still happening, but they don't grow. So it's the only thing that both papers have. So based on this, what I'm working on is creating hypotheses of what I think it's actually happening in Sargassum. So for example, for Sargassum fritans, I know that it has higher growth rates because it's the morphotype we see the most. Then that it will have an optimal temperature of 28. Sargassum natans, they told you this decoupling between photosynthesis and growth means that Sargassum it's not allocating energy to grow, but it's still doing photosynthesis. So it has higher maintenance rate. That optimal temperature, it's lower temperatures. This is the morphotype we see the less when they do abundance per morphotype, when you study abundance per morphotype. And the only thing I have about Sargassum natans vincae for now is that it has higher photosynthesis efficiency. But so this is something I have to work now to do simulations with this hypothesis to see if I'm able to explain the results they are having. The third part of my PhD then is the dispersion model. I think you know Christophe Lett, Nicolas Barrier, and I don't know if you know Vittel. He was here doing his PhD also in Sargassum. And he creating this in vitro lab with the simulations. He has all the codes developed already to run simulations on Sargassum. So this is something I will not take from him, but I will use I will use his simulations and his setup. I will only change the initial conditions. And then I will try, for the first thing I will do is from the trajectories we see here to extract the environmental conditions, adapt my model, because for now I had a model that was for lab conditions. Now I have to adapt it to the oceanic scale. and to run simulations offline and compare both the results of beetles and my results to see if different models can get similar results or what is happening now. Now, what is happening? So this is what I have started doing. The power of DEB is that before I show you, we had only stable conditions, but now we have dynamic conditions because we're in the ocean. So every time step, we have different light, different temperature. So here with DEB, I'm able to capture this. This is the simulations I did with the trajectories I showed you in the last slide. So I did the same work I did for the DEB model for Sargassum in constant conditions. I translate the model oceanic environmental variables to forcing variables in my abstract world, that is the depth forcing variables. I did the equations and I translated the state variables into what it's measured, for example, growth, photosynthesis, and the CNP ratios. And now I can study actually what's happening inside of the algae, how much photosynthesis is he producing, and what. The next step, and something maybe I will not have the time to do because I only have 1 year left, it's to study the internal variation. As I told you, now we can study the seasonal variation, but we don't know what's happening if the sargassum of last year, for example, it's feeding the sargassum of next year. And we would like to test this, but we still have a lot of questions about this. There's question about where does sargassum come from? Does it come from the Guinea Sea? Does it make like the whole, does it turn like this? And we have questions about this, and we have to still work about this question. So as the conclusion, we have a depth model that for Holopelagic Sargassum that it's now working and that we can use to test different hypotheses. We also have an approach to guide experimental studies. And from this, there's a paper in preparation for my PhD. Then for the three morphotypes, we see that there's no clear difference, but we can make hypotheses, and this is the power of modeling, is that we can make hypotheses, test, okay, it doesn't work, okay, I will test another thing and continue and continue. And for the year-to-year simulations, we can do it, we can compare it, it's just that we have to do it. And then we have these questions, as I told you, the question marks of what is happening if the sargassum of n-1 is feeding sargassum of n. And so if it's maybe survival processes, there's a lot of questions here that we still have to think about. So thank you.
Thanks, Maria. We can start with questions online if there are any. Florencia, oui.
Hello, nice talk. Thank you.
Hello.
I was wondering, so when you towards the end that you mentioned about if they're traveling from the coast of Africa to Caribbean. So do you incorporate like oceanography or like currents or something like that into the model, or how do you, or is it actually part of your questions like the movement of them? Or
It's not part of my question because this is the question that Witold already worked on. So his PhD was about this. His PhD was about if it was, for example, what wind factor and which currents would have. I think he also put the Ekman into the model to see if he could explain only the transport. So that's why I'm using his codes to only change the growth model he already used to see if we have different results, because his model was a Eulerian model, so mine it's a Lagrangian model. And we will see if having this and also having the capacity to have the different reserves help us better explain what we are seeing.
right, okay. Right, thank you.
Thank you.
There was another question, Tim, online.
Yes. So thank you. So it was very, very interesting your talk, and congratulations, because it's a huge work. And yeah, my question was about the interesting method that you developed to estimate the number of data you may need to constrain your model. Um, I think it's a nice method that you developed. It's an empiric method, and I wonder, do we have in the DEB in DEB formalism, can we have also a theoretical estimation of the number of data needed to constrain your model?
Mm.
If you get my question, I can. Otherwise, I can precise.
I'm not sure because this is an empirical way, but it's actually all in the theoretical because it's all in the framework of my model. I have not in this type of protocol. I don't have real data. The real data was generated.
I understand that, but I mean empirical in the sense that you are doing numerical experiments each time to test if it works, if it's sufficient number of data or not, if I understood well. But what I mean is, for example, if you have a system of two unknown x and y, you need two equations with different values of the parameters, and then you can know the values of x and y. So I wonder, can from the number of parameters you need to estimate for your DEB model, isn't is it possible, or do you know if there is theoretical works that exist that can tell you in advance how many observations you need in different environmental conditions to constrain your model?
But it's sort of the work I'm trying to do, but there's also other papers, especially for DEB, that have tested this also in other cases. They use more powerful statistic measurements that I do because I did a very simple test. It's just that my model is more complicated than the model they use. So I have a lot of processes to test and a lot of parameters in this model, and they use less parameters. But they have studied this also to have, for example, what kind of data and even the number of points in one curve. If only having the initial and final it's enough, or if we need more data points across time, for example. So yeah, it exists and it's possible.
Okay, thank you. Congratulations again.
Salle. Nicolas, vas-y.
Oui, vous m'entendez? Bonjour Maria.
Bonjour.
I have a question about the calibration you mentioned and the calibration issues. I have not understood what was the variable you used for the calibration. You calibrate on what?
So I have So I have my data. This is the data available I have. And what I did for this method is that I generated, so I have the environmental conditions they use, and using the environmental conditions and a set of parameters that I call my reference
Isn't it
parameter.
before? This is the numerical experiment. This
Yeah,
one.
this.
But before, yes, before you said that the calibration was failed because you had not enough data, but I have not understood on what criteria you made the, not criteria, on what variable you made the calibration. You calibrated
On this data,
which are
which
the
is
weight.
wet weight according to different temperatures and photosynthesis rate.
So you try to set or to calibrate the depth parameters so that you recover the wet weight.
Yes.
Okay.
Yes.
Okay. Okay. And we don't, sorry if the question is stupid, but
No.
we don't see the amount of data you have. We see regression and days. Why you have plenty of weights on which to calibrate.
But it's not the only data I have. It's these 2 points. So for
Okay.
each data set, I only have the points, and
So
okay. And the lines. It's my calibration. It's the result of my calibration.
Okay. So there are
Yeah,
plenty
sorry.
of sargassum, but only 4 points.
No. And actually, I don't know if anyone has seen the other DEB papers, but when you compare with the fish, for example, where you have points like this, and then you have your curve, that it's amazing. But I have to the problem is this, that I don't have enough
Okay,
data.
okay, fair enough.
And yeah.
Okay, thank you.
This is something that has not been studied. So I don't know, even fragmentation, it's so weird. Apparently there's in Thierry Thibault in Marseille told me that Sargassum dries because of the sun, then it breaks, and from this broken part, it restarts. Yeah. It's amazing. Like, how is it possible? So there's no lab studies about this. And there's Yes, but it's taken into account. In the DEB model, we have the sites of the organism that is taken into account. Yeah. It's not the same, and because and maybe it's also, for example, for fragmentation, each morphotype it has a different elemental composition. There's one of the morphotypes, and I think I think it's Vinge, that it's more rigid than the other one. So I think this will affect also the floatability of the organism. And in the case of the lab conditions, we don't take into account this, but in the dispersion model, the depth of the particle it's taken into account as if it passes, for example, 100 meters. We can consider that sargassum cannot go back to the surface, and it just will only sink.
Designing, like, for example, read.
Yes. Yes, that's right. And there's Paulo Duarte Neto in University of Brazil. He's trying to work on the formation of these morphological parts of sargassum. So, for example, here we have the leaf, we have the stipe, and we have the air bladder. He's trying to work on this. And one of the objectives of the project was to have DEB combined with the morphological creation of the parts of it. But it's harder than we thought to do this. Even for otoliths, it's so hard to make the combination of both. But yeah, it's true.
Is there another question online, maybe? Olivier?
Yes, hello everyone, and hello Maria. Are the morphotypes independent? I mean, can they turn into each other? Can one morphotype transform into another one?
No, as they only reproduce by fragmentation. Each morphotype, each sargassum that we find in the sea, it's a clone of the other one, and they cannot mix between both of them. It's really and genetically they are separated. So
Okay, so you can consider them as competing, interacting species.
But yeah, I can consider them because they are all floating at the same time. So there's fritans and nattans together, so maybe they are competing for the resources, and this is something we can think about, because if they have different, for example, assimilation parameters, then it means that one of the morphotypes is taking more than the other one. But this is not like I will not work in the competition between morphotypes, but it's something that we can also think about, that it's happening at sea.
Okay, thank you.
Thomas?
Yes, I have a question. It's
Hello.
about whether you can include the diazotrophic assimilation pathway into the DEB model. You know, in the wild, the sargassum, there are many. They're living with many other organisms which can fix the azote in ammonium form. So
Yes.
the physiology is based on it may be based on symbiotic relationships. Can you include that in the DEB model or not?
I think if we want to include it, the first step will be to consider how much nitrogen it's actually going from the bacteria to sargassum, and then include it in. So if we see the model here, we have different kinds of nitrogen that are going in. We have nitrate and ammonium. So I think the first step will be a step that we can make is to consider, okay, N2, how can we, if we can measure it, put it as a quantity, a concentration here that goes into the algae. And I also think that it will be helpful in this case to consider also that if they have more things attached to their body or the algae, they have less light coming in. So there's this hypothesis that we can make, like it's called overshading when you have different organisms that are one next to the other. But also for sargassum, this is something we can think about. But
Because I just have a suggestion for this question, because you know that the atomic ratio is not the same when it is the azote is come from the diazotrophic pathway. So you have an isotopic ratio which can identify the origin of the azote in the composition of the sargassum, of course. So
okay.
I think it's a way to make the discrimination between what should be azote coming from, I should say, a direct pathway or through macrobiotic organisms.
Okay.
So this is the isotopic composition of azote of nitrogen is very important to take into account for that.
Yeah. Because actually, also sargassum, it passes through a lot of oligotrophic zones in the Atlantic, and this is one of what we think in the sargassum community is that they live because they have this nitrogen coming from the bacteria. So it will be interesting because maybe that's what's happening. That's why they are living and proliferating in the Atlantic. But for now, I think the model from Joanno did consider this atrophic. I don't remember if they consider it, but it will be interesting.
Thank you for the answer. Thank you.
Thank you. Yes, you cannot. For one of the data I have to constrain my model is, for example, CNP ratios, the quantity of carbon, nitrogen, and phosphorus, and this comes from data at sea. And this is a specific one and only point. But you can also use if you manage to follow a sargassum and do different measurements, yeah, you can have it. But for example, in other cases for fish, they do have data from the ocean, for example. So you can also integrate them in the estimation.
Okay, so if there's no more questions, thanks you again.
Thank you very much.
Thanks everyone online, and see you next week.
Ça marche.